Hello, my name is Jorge Gomez, and here I will be discussing the work that I am currently doing in Dr. Lynette Dawes' lab entitled Investigating the Serotonin Transporter and Organic Cation Transporter 3 in Serotonin Clearance in Basolateral Amygdala and its implications for stress-related disorders. The natural question that arises is why study serotonin neurotransmission in the basolateral amygdala? Well, when serotonin neurotransmission is disrupted, we know this leads to a variety of stress-related psychiatric disorders. One of these disorders being post-traumatic stress disorder, or PTSD, which is highly comorbid with drug abuse. We know the primary way of clearing serotonin from the extracellular space is the serotonin transporter. However, our lab has shown that in the hippocampus, the organic cation transporter 3, OCT3, is also important in regulating serotonin clearance. Additionally, OCT3 is highly expressed in the basolateral amygdala, thus making it an important structure to study serotonin uptake. The technique we use to study serotonin neurotransmission is in vivo chronoamperometry. The way this works is by having a Nafion-coated carbon fiber electrode attached to a multi-barrel pipette, which we use to pressure eject serotonin with the different drugs we are testing, and we measure the amplitude and the clearance rate. As you can see here on the far right, our preliminary data shows that blocking OCT3 with D22 augments the ability of the SERT blocker fluvoxamine when compared to either blocker alone. However, D22 blocks other OCT subtypes and the plasma membrane transporter, thus we decided to use the more selective OCT3 blocker, corticosterone. As you can see here on the right, similar to D22, corticosterone potentiated the ability of fluvoxamine to inhibit serotonin clearance in a manner that appears to be independent of corticosterone's actions at glucocorticoid or mineralocorticoid receptors, as you can see here in the first three figures. Taken together, this data suggests that dual inhibition of the serotonin transporter and OCT3 enhances serotonin actions in the basolateral amygdala. Going forward, we will continue to increase the sample size to finish off these experiments. We will also use optogenetics to dissect the circuitry involved with serotonin transporter and OCT3 in the serotonin clearance in the basolateral amygdala and its associated stress-related behaviors.